Hello and welcome to this special series on biology. We will be discussing biology from a very basic level and then we will graduate on to more complex issues. We will start by discussing various themes of biology. Studying biology is a difficult and uphill task. There is so much of diversity of life on our planet. Biologists have so far identified and named about 1.8 million species. There is so much amount of information and the branching out of the various fields can be compared to a very intricate and complex maze. And of course, without any proper division of areas or subjects, one can simply get overwhelmed and lost. So we have tried to arrange this humongous data into different themes, which can have topics of relatively unifying content. Now, let's talk about emergent properties. It is the arrangement of elements which can be broken down to cellular or molecular level, which explains the working of different parts of an organism. For example, if you take chloroplast of a green leaf and put it in a container with water, nutrients and air, nothing will happen because chloroplast works in a specific arrangement in a cell and it interacts with sunlight, water and nutrients to start the process of photosynthesis. It is like if we put all parts of a car in a box, it would not result in a moving car. A moving car works only if all the various parts of a car are arranged in a specific and synchronized manner. The emergent properties are a useful way to understand the various complex arrangements in biology and can be applied at molecular level to the bigger scenarios like ecosystem. On the other hand, taking out the components in an organism, separating it and studying it in isolation is also essential to understand its working and properties. This isolation of components for studying is called reductionism, which is the reduction of complex systems to simpler components that are more manageable to study, can become a powerful strategy in biology. For example, the concept of genetic and heredity was understood when James Watson and Francis Crick in 1953 were able to identify the DNA, that is deoxyribonucleic acid molecule, and inferred how this molecule could serve as a chemical basis of inheritance. The central role of DNA in cells and organisms became better understood. Biologists must balance the reductionist strategy with a larger scale holistic objective of understanding emergent properties, how the parts of cells, organisms and higher levels of order such as ecosystems work together. In biology, a system is simply a combination of components that function together. A biologist can study a system at any level of organization. A single leaf cell can be considered a system, a tree, a rat or a marsh ecosystem. In system biology, models are constructed for understanding the dynamic behavior of whole biological systems. Successful models enable biologists to predict how a change in one or more variables will affect other components and the whole system. How a drug that helps control pain affects the other parts of body, or how an increase in carbon dioxide in atmosphere can change ecosystems. The cell is the most elemental and basic unit of studying biology, as it contains all components for sustaining life on its own. Understanding how cells work is a major focus of biological research. All cells share certain characteristics. For example, every cell is enclosed by a membrane that regulates the passage of materials between the cell and its surroundings. And every cell uses DNA as its genetic information. Now we will talk about two different categories of cells: prokaryotic cells and eukaryotic cells. The cells of two groups of microorganisms called bacteria and archaea are prokaryotic. All other forms of life, including plants and animals, are composed of eukaryotic cells. A eukaryotic cell is subdivided by internal membranes into various membrane enclosed organelles. In most eukaryotic cells, the largest organelle is the nucleus. It may be vacuole as in plant cell, which contains the cell's DNA. The other organelles are located in the cytoplasm, the entire region between the nucleus and outer membrane of the cell. Prokaryotic cells are much simpler and generally smaller than eukaryotic cells. In a prokaryotic cell, the DNA is not separated from the rest of the cell by enclosure in a membrane bounded nucleus. Prokaryotic cells also lack the other kinds of membrane enclosed organelles that characterize eukaryotic cells. Now, we talk about the building blocks of life, that is, chromosomes. The chromosomes have almost all of the cell's genetic material. It is made up of genes, the units of inheritance that transmit information from parents to offspring. Each chromosome has hundreds or thousands of genes arranged along its length. Each of us began life as a single cell stocked with the DNA inherited from our parents. When a cell replicates, it transmits a copy of the entire DNA in the offspring cells, and this process carries on till the required organ is created, involving constant replications resulting in trillions of cells. In each cell, the genes along the length of the DNA molecules encode the information for building the cell's other molecules. In this way, DNA controls the development and maintenance of the entire organism. The DNA serves as a central database. Each DNA molecule is made up of two long chains arranged in a double helix. Each chain link is one of four kinds of chemical building blocks called nucleotides, namely adenine, that is A, cytosine, that is C, guanine, that is G, and thymine, that is T. We can think of nucleotides as the alphabet of inheritance. Specific sequential arrangements of these four chemical letters encode the precise information in genes, which are typically hundreds or thousands of nucleotides long. The production of cells, proteins, hormones, enzymes, organelle, etc. is triggered by the genes responsible for it. So, DNA provides the blueprint and the proteins serve as the building material that actually create and maintain the cell and carry out its activities. The DNA of genes controls protein production indirectly using a related kind of molecule called RNA, that is ribonucleic acid, as an intermediary. The sequence of nucleotides along a gene is transcribed into RNA, which is then translated into specific protein with a unique shape and function. In the translation process, all forms of life employ essentially the same genetic code. Thus, DNA is the coded or stored information, whereas RNA is the processed information. DNA of every cell is same, whether it is a skin cell or heart cell or brain cell, whereas RNA of the cells varies and hence the differential expression of proteins is seen in different cells. This is called as central dogma. RNA molecules also catalyze biological reactions, control gene expression and communicating responses to cellular signals, and are essentially of three types: messenger RNA, ribosomal RNA and transfer RNA. Protein synthesis, a universal function whereby mRNA, that is messenger RNA molecules, direct the assembly of proteins on ribosomes. This process uses transfer RNA, that is tRNA molecules, to deliver amino acids to the ribosome, where ribosomal RNA, that is rRNA, links amino acids together to form proteins on the template of DNA. The entire library of genetic instructions that an organism inherits is called its genome. A typical human cell has two similar sets of chromosomes, and each set has DNA totaling about 3 billion nucleotides. Within this genomic library of nucleotide sequences are genes for about 75,000 kinds of proteins and yet unknown number of RNA molecules. The entire sequence of nucleotides in the human genome or DNA is now known. The supercomputers and the development of new methods and DNA sequencing machines has made it possible. But this is just a beginning, as the effort to learn how the activities of the different proteins encoded by the DNA are coordinated in cells is underway. Now here we have to apply the system approach at molecular and cellular level as discussed earlier to sort out the huge data generated from the sequencing project. This figure illustrates the results of a large study that mapped a network of protein interactions within a cell of a fruit fly. The model is based on a database of thousands of known proteins and their known interactions with other proteins. For example, protein A may attach to and alter the activities of proteins B, C and D, which then go on to interact with other proteins. The figure maps these protein partnerships to their cellular locales. This diagram maps 2346 proteins represented by dots and their network of interactions, lines connecting the proteins in a fruit fly cell. Systems biologists develop such models from huge databases of information about molecules and their interactions in the cell. A major goal of these systems approach is to use the models to predict how things change, such as an increase in the activity of a particular protein can ripple through the cell's molecular circuitry to cause other changes. The total number of proteins in this type of cell is probably in the range of 4000 to 7000. The system approach can be applied to this example in following way. First, it is important to list as many parts of the system as possible, such as all the known genes and proteins in a cell. This is an action of reductionism, that is, breaking down of complex systems to simpler components that are more manageable to study. Another important theme in biology is feedback mechanisms that regulate biological systems. Like for example, when you do a strenuous exercise, the demand for sugar in your body shoots up and more sugar lands up with the muscles that acts as fuel, and in case the body is at rest, the sugar is metabolized by a chemical process and stored. Now these chemical processes happen or are catalyzed with the help of a catalyst agents called enzymes. Each enzyme catalyzes a specific chemical reaction. Now the question is, how does a cell coordinate its various energy or nutrient needs? In our example of sugar management, how does the cell match fuel supply to demand, regulating its opposing pathways of sugar consumption and storage? The key is the ability of many biological processes to self-regulate by a mechanism called feedback. In feedback regulation, the output or product of a process regulates that very process. In life, the most common form of regulation is negative feedback, in which accumulation of an end product of a process slows the process. For example, the cell's breakdown of sugar generates chemical energy in the form of a substance called ATP, that is adenosine triphosphate. When a cell makes more ATP than it can use, the excess ATP feeds back and inhibits an enzyme near the beginning of the pathway to stop making more ATP. There is also a positive feedback in which the end product speeds up its production. The clotting of your blood in response to injury is an example. When there is a leakage from bloodstream due to any reason, the defense mechanism in the blood comprising of various proteins referred as factors are activated in a cascade to stop the leakage by formation of a clot. Feedback is a regulatory motif common to life at all levels, from the molecular level to ecosystems and the biosphere. Such regulation is an example of the integration that makes living systems much greater than the sum of their parts. Now, to summarize, we have started to discuss the basic themes of biology and have briefly touched the various aspects of life and the basic mechanisms. We have discussed about concepts like emergent properties, system approach, reductionism, etc. And understanding of these terms would make the process of understanding the various complex processes in biology simpler and easier to comprehend. We have touched upon the process of molecular biology and DNA, which we will study in detail, and also we have tried to understand the process of feedback in which the body learns to regulate its mechanism, energy needs and nutrients right down at cellular level. Thank you so much for watching. I hope this was a learning experience for all of us.